Thank you so much, Joe, and Joe and Joe, and thank you all so much for joining today's webinar. I'm really humbled to be here, presenting to you all and presenting and sharing my science. Yeah, there's my contact information, and you'll see it at the end if folks would like to reach out following this webinar. Now, I want to start off this talk just like 20,000, 30,000 view, kind of this blue sky idea. Because when I step back and I think about a unifying thread in the environmental sciences, I think a theme that keeps popping out is this idea of heterogeneity in environmental systems, or how things are different over space as well as over time. And many of us, myself, I work a lot with satellites, and it's kind of easy, or it's more easy to see and appreciate how satellites help us find this heterogeneity, tracking changes over large spatial extents and over decades even. But I think many of us are chasing that sort of like bigger question of not only identifying changes or those heterogeneities, but ultimately why we're seeing those heterogeneities or contextualizing them. Now, that's all kind of abstract, so let's try to make that a little bit more concrete. Now, at the U.S. Geological Survey, many folks are very familiar with the stream gauging networks. And here I have a map of the contiguous U.S., where these blue filled in dots in the eastern United States are associated with above average stream flows. And these open red circles in the western US are associated with below average stream flows. And I think what this picture shows this picture is a really nice example of this idea of heterogeneity. We're seeing differences in stream flows in two different parts of the region. But we're also able to contextualize those heterogeneities when we bring in external data. And so, for example, the eastern U.S., we see frontal systems bringing in high water to the eastern U.S., giving rise to these elevated stream flows. We see extreme drought persisting in the western U.S., giving rise to these below average stream flows. And I think there's been a lot of really excellent work that's been done over decades, working with these sort of macro scale water quantity questions. And I think that we're really entering this new era of working with water quality and getting at these water quality questions at larger scales. And a great example, I think, of this is this work by Ben Kramer, who used, Ben, I believe he's at University of Freiburg now. He used the MODIS satellite to look at trends in greening and bluing, so chlorophyll trends, in several of the world's largest lakes. And what we can see here is that, heterogeneity is ever present. We see that in many cases, like Lake Michigan, Lake Baikal, we're seeing greening in the nearshore area, bluing in the offshore, but there's also places like the Caspian Sea where we're seeing this greening in the northern part of the Caspian Sea, bluing in the lower part, and some mixed heterogeneities in that central basin. So just keeping with that theme of, we're starting to be able to really look at these heterogeneities across systems and within systems and over decades, but as it pertains to water quality. Another example that I'm involved in as a co-I is the cyanobacterial assessment network. It's really spearheaded by the US EPA and is a really grassroots yet expansive network across about 5 federal agencies. And what this work effectively does, it allows users in sort of this point and click interface to get a general assessment of cyanobacterial abundance for various lakes within the contiguous US. So here we have a picture of Lake Mendota. Sorry, I live in Madison, Wisconsin. Mendota's just right over there. So I had to throw that one out there. And what we can see is that through this use of the Sentinel satellites, that's through the European Space Agency, we can combine in situ data with satellite remote sensing imagery and ultimately track changes in cyanobacterial abundance. So here, in this time series on the left, we have these bar charts, and we can see that those orange high cell counts were pretty high in August of 2024, and then it transitions to medium and low cyanobacterial abundance through time. So ultimately, I think tracking this sort of phytoplankton successional stages through time, at least as it pertains to cyanobacteria. So these are the types of questions that we're starting to get at with satellites. And one other example that I'm involved with that's not available to the public is this remote sensing of water quality dashboard tool that we're developing at USGS. Here, an example of turbidity at the confluence of two rivers in Philadelphia. And what we can see here is that theme of heterogeneity is ever present. We have this bright green turbid water on the western side mixing with this blue-green turbid, this blue-green water on the eastern side that's supposed to represent a high and a low turbidity. And what we can imagine is that if I'm a field tech, this tool is really designed for the technicians, where you're placing a sensor in a river, especially where there's a confluence, might give you a skewed perception. Yeah. And so we're hoping that these satellite tools can not only help us understand across ecosystems, but also what water quality concerns might be happening within ecosystems. And I'm a lake person, but I had to throw in at least one river example that we're working with. So that's broadly what's happening. Where does my work or my core work fit in with this? And as the title suggested, I think a lot about the idea of trophic state or lake trophic state. Now, that's kind of a niche term, maybe, for many folks. So if you're less familiar with this term of trophic state, I just want to walk through what I'm talking about. And we generally think of trophic state as this idea of how productive are lakes. Do they emit more carbon than they retain? Do they have more algae or more things eating algae? Stuff like that. And we generally think of lakes as being oligotrophic or eutrophic. You can think of an oligotrophic lake as being bright blue, low algae. This is a picture of Flathead Lake in western Montana. It's the type of lake that you want to swim in. Then there's eutrophic lakes. This is Lake Mendota on a particularly smelly day. And it's bright green. There's lots of algae. It's kind of stinky. It's not the lake you want to swim in, but the lake you might want to fish in. And that's generally what we're talking about when we're talking about oligotrophic and eutrophic. Now, those words alone, if you take like a limnology 101 class, classifying a lake as oligotrophic or eutrophic comes with a lot of knowledge, decades of knowledge. And we can sort of like infer these generalized expectations for how these ecosystems function. And a great example of this is how temperature and oxygen profiles or characteristic oxygen and temperature profiles shake out when a lake is stratified in the middle of the summer. And these are actually real data collected from the US EPA's National Lakes Assessment, where we see oligotrophic lakes in the summer should have this characteristic temperature curve where it's high at the surface and it sort of tapers off and then becomes pretty constant below some depth. Oxygen should start high at the surface, then it should dip down to the point where we're seeing that inflection with temperature and then start to increase towards the bottom of the lake. In contrast, eutrophic lakes in the middle of summer should have a similar temperature profile, but the oxygen, when it starts high at the top or high at the surface, decreases till you hit some inflection point and then remains constantly low until you reach the bottom or the bottom of the lake. And these are things that you learn in Limnology 101, but they're the types of processes that we can easily connect to each of these classifications. Once we say a lake is eutrophic, this is the temperature oxygen profile that a limnologist would intuit. Now, when I'm talking about trophic state, I'm actually thinking a little bit more broadly, going all the way back to like the 1800s of how trophic state was originally thought of. And it's really thinking in this bivariate axis, where we here on the x-axis we have total phosphorus, so that gets at that that algal component of the lake, and on the y-axis we have true color, and that's really some proxy for how much organic matter or how brown is your lake. And this is called the nutrient color paradigm. And it's with this paradigm that we can actually break lakes not into those two groupings of oligotrophic and eutrophic, but in four groupings. And so we can call lakes oligotrophic if they have low phosphorus concentrations and low true color metrics or low organic matter. Eutrophic or green if they're high phosphorus, low organic matter. Brown or dystrophic if they have low phosphorus, so low algal productivity, low zooplankton, low higher consumers, but high organic matter. And then mixotrophic or murky if they are combinations of both high phosphorus, high nutrients, and also high organic matter. And, it's this framework that I think has kind of gone through this Renaissance period with Craig Williamson's work in the late 1990s. Katie Webster from Michigan State has a great paper in 2008. And Dina Leach in the top right, Stephanie Hampton, Amina Pollard, and Steph LeBoux led a really great assessment using the EPA's National Lakes Assessment to look at changes in how the U.S.'s changes in lakes of each of these four categories. And what they found is that nationally, blue lakes tend to be decreasing. Green lakes, those eutrophic lakes, tend to be about the same. And brown lakes and murky lakes, these high organic matter lakes, seem to be increasing. But getting back to that theme of heterogeneity, that national trend is really driven by a few key regions or ecoregions. There's the Northern Appalachians, like the Northeast. The Upper Midwest has a fair amount of browning. Southern Plains has some murkifying and xeric environments, some murkifying lakes. But this is just, this was really that first emphasis, or one of the main emphasis, let's say that, of looking at national trends or national changes in these lake trophic states. But many other folks have been kind of getting at this question just with slightly different framings. Simon Topp is a great example of this. He used the Landsat record to look at changes in lake color using this metric called dominant wavelengths. And Simon basically showed that, there seems to be decreasing green lakes moving into bluish lakes through time. But it's just another way of assessing these changes, and it's kind of creating this alternative viewpoint, or this kind of bit of a head scratcher as to why these changes are happening. I'm happy to talk about that later. But that's for a bunch of lakes over a bunch of time points. And it's also observing changes. There's also been a lot of work looking at ultimately contextualizing that change. And Bella Alexa at CU Boulder is one of my colleagues that's also been thinking about this a lot, and working with a handful of lakes where we have really good environmental data. And Bella's work has been showing that hydrologic settings, so things like the residence time of a lake, hydrologic connectivity, climate variability, has been really important for ultimately governing why these lakes might be transitioning between between blue, green, and brown. But it's not just Bella that's been working on this. There's a lot of people asking why these changes might be occurring. And, frankly, if we wanted to pinpoint a mechanism, it'd be hard to always say which one might be at play, because there's a bunch, and many of them are disputed. So Bella's work looking at hydrologic setting, there is a wealth of literature showing that acid rain has contributed to lake browning, particularly in the northeastern United States. And even things like invasive species, things like zebra mussels, quagga mussels, have been shown to change lakes from green to blue. And so there's a bunch of different mechanisms that could be at play, especially at the continental scale. And so if we wanted to get, or the national scale, let me say that, if we wanted to get at these sorts of questions, we would need a very large data set that looks at multiple different lakes over multiple different years and has multiple different drivers associated with each of those lakes. And so we made that. And we did that by merging between remote sensing data and in situ data to really get at this idea of trophic state. You can scan the QR code to access our scientific data paper, but I'm just going to condense how we made this data set into a few small steps. First, we identified parent data sets that we could use to generate this data set. And so we combined the 3 years of the US EPA's National Lakes Assessment with limnosat, which is this aggregated Landsat surface reflectance data set for about 55,000 lakes from 1984 through 2020. And Simon Top is the person who developed that data set. And each of those aggregations involves the use of hydrolakes, which is this compendium of lake locations and morphometric data for each of those lakes within limnosat. And so we can basically take this combination of in situ data, reflectance data, and locational data in tandem with our trophic state data set to create, definition of trophic state, to create a harmonized training data set where we know the in situ classification and we have the associated surface reflectances from the satellite, and we can create different classification models to relate between those two. So what we did is we used that training data set to create different machine learning, neural network, and even parametric, classic statistics models to create this sort of ensemble of predictions for lakes where we know the trophic state. And we spent a lot of time validating those classification models, and you can read all about those details in the scientific data paper. Suffice it to say, we have a really good handle on when the models make a bad classification and what those types of what those factors that drive the misclassification really are. But we can take those pretty well validated models and apply them to lakes where we don't know the in situ trophic state, and for years when we don't have in situ data for that trophic state classification. And so what that allows us to do is to create a time series of trophic state classifications for all of these lakes within the contiguous US. Well, so there's always a chance that I did a really bad job of explaining all of that. And eyes might glaze over. So if that describes you, suffice it to say, we took the approximately 1,600 lakes that are within the US EPA's National Lakes Assessment, merged it with Landsat surface reflectances, created that classification model, and then applied those models so that we can have estimated trophic state or predicted trophic state classifications for about 56,000 lakes throughout the contiguous US. So going from this map to this map. And it's with these data that we can ask pretty fundamental questions. Things like, how has lake trophic state changed nationally? And so what I'm going to show on this next slide is a time series. Year is on the x-axis. Y is the proportion of lakes of that trophic state through time. And what we see is that green lakes since the 1980s seem to be decreasing somewhat slowly. Blue lakes tend to be decreasing since the 1980s, but these brown lakes seem to be especially increasing all the way since the 1980s to, the 2020 till 2020. But it's not just it's not just the national trends. Like I said, this idea of heterogeneity. We can also ask questions like, where are these lakes? Oh, sorry. And I want to also add, the models only know the red, the green, the blue, and the near infrared bands from Landsat. They do not know where a lake is. They don't know anything about the lake or the year or anything. They just know the colors of the lake. And so what I'm going to show on the next slide is a map of the US, and each dot is colored by the proportion of lakes of that trophic state in that basin. So like a brighter color means more lakes of that trophic state. And so if we ask, where are those oligotrophic lakes? Here we have these bright blue dots, right? Like I said, brighter blue means more oligotrophic lakes. And what we see is not entirely surprising. Oligotrophic lakes tend to be concentrated in the Northeast, in the upper Midwest, the Pacific Northwest. They follow mountain ranges. Generally where we would expect oligotrophic lakes. Eutrophic lakes tend to be concentrated in the Great Plains, in the Southeast, in the southern United States, and notably not where we have oligotrophic lakes. Now, dystrophic lakes, and I just want to remind folks, the models only know the color, not where the lake is. Dystrophic lakes tend to be concentrated in the upper Midwest and the northeastern US. As the previous plots showed, these lakes tend to be much less common than the other two lake types, but they seem to be increasing through time, and they seem to be concentrated in these two ecoregions. And what I also want to note is that not only can we look at how or where are the lakes, but also how are they changing in each of these basins. And we see that oligotrophic lakes, if we were to take the slope of the proportion of change through each of those basins, we see that oligotrophic lakes their change is pretty heterogeneous over at continental scales. We see some things where in Michigan and Minnesota, to some extent the Northeast, we're seeing increases in oligotrophic lakes. That seems to correspond with a lot of in situ efforts that are going on in these regions. But there's these areas like in northern Minnesota, northern Wisconsin, the Adirondacks, the Northeast, we're seeing decreases in oligotrophic lakes. Eutrophic lakes, we're seeing generally decreases in many of these ecoregions, where increases are pretty heterogeneous throughout the US. But those dystrophic lakes are particularly increasing in the upper Midwest and the Northern Appalachians pretty uniformly. And I'm thinking of these areas as largely being hotspots of this ecological and limnological change. Now, going back to how Dina Leach aggregated the National Lakes Assessment data, we can also think about how each of these ecoregions that the EPA tends to monitor or tends to think about are likewise changing. And so we can look at that proportion of change within each of these ecoregions, and what we see is that by and large, most of the ecoregions are pretty constant through time, with the exception of the Northern Appalachians and the upper Midwest, so kind of that theme that we've been thinking of. And what we're seeing is that generally in these regions, the eutrophic lakes seem to be decreasing since the 80s. Oligotrophic lakes seem to be pretty constant. And the dystrophic lakes, those brown lakes, are increasing. So still keeping with that same theme, no matter how we spatially aggregate those data. But what we frequently get asked is, well, are lakes switching from green to brown? What's going on? And when we track individual lakes throughout this time series, we see that, by and large, there's a fair amount of variation between blue lakes switching to green and green lakes switching to blue, but those brown lakes, they tend to be that are concentrated in the Northern Appalachians and the upper Midwest. Those tend to be areas where those brown lakes are almost exclusively coming from blue lakes. And if you remember, those blue lakes were pretty constant through time, but the eutrophic lakes were decreasing. So what this implies to me is that we have, in these two ecoregions, we have green lakes switching to blue lakes switching to brown, but those rates of change are kind of canceling each other out, which is keeping the proportion of blue pretty constant through time, and leading to a decrease in green and an increase in brown. So I just threw a lot of plots and a lot of words at you. So let's just step back for a second and think and try to synthesize some of these ideas. Dystrophic lakes, they seem to be increasing in the upper Midwest and the Northeast. They seem to come from blue lakes. Other regions seem to be not changing. And the upper Midwest and the Northeast are ultimately those regions that are driving national trends, likely because there's so many lakes in those regions. Eutrophic lakes seem to be declining in the upper Midwest and the Northeast, and they come from and transition to blue lakes. And then oligotrophic lakes, at least at the ecoregion level, seem to be remaining somewhat constant, but they seem to come from green and become brown. They might also switch back to being green. Now, getting back to that main theme, we've identified these changes, but what about contextualizing that change, or what's the mechanism, Michael? And so to do that, we aggregated a bunch of data for all of these lake basins. And I just want to walk through what exactly we did. I think the best way to do this is looking at it by the numbers. Excuse me. So for about 38,000, 39,000 lakes located in about 15,700 unique basins throughout the contiguous US, we aggregated a bunch of drivers that the scientific literature seems to suggest might be driving these changes. And to be quite clear, how did we aggregate these drivers, or how did we think to aggregate these drivers? Well, we read about 330 papers in the scientific literature and found what available data sets might be available at scale to address these sort of hypotheses. And so to get at potential hydroclimatic drivers, we use temperature and precipitation data from NASA's climate reanalysis data. We use snow area from the MODIS satellites, modeled evapotranspiration from the global lake evaporation data set. We calculate PDSI from climate reanalysis as well as remotely sensed data. We aggregated the entire NADP record for sulfate, nitrate, ammonium, and H+ deposition. We used all of the NLCD land cover classifications to look at land cover type, as well as Oak Ridge National Lab soil organic carbon product, as well as the UK Center for Ecology and Hydrology's soil phosphorus product. For invasive species data, we used the USGS's aggregated invasive species catalogs that has at least where invasive zebra mussels and quagga mussels were identified within the contiguous US. Certainly not a complete data set, but the best that we have at scale. Lake-specific data, a lot of this came from hydro lakes, and to get at seasonal fluctuations in lake surface area, we used the Landsat record to look at seasonal water surface area as well as permanent, and also to look at ice cover, we use the MODIS ice product for each of these lakes. And so basically what we do is we take you can think of a giant Excel spreadsheet where we're trying to relate the proportion of a given trophic state in a given basin to all of these different variables. And we throw it in this random forest model, and we evaluate how the model associates each of those variables with a given proportion, with a given amount of lakes of a given trophic state. So that was kind of a word salad. So effectively what we're able to do is use these models to understand which of these variables are most associated with increases or decreases in trophic state. Now what I'm going to do is, looking at time, I'm going to show some regression trees, and they're kind of ugly. So I'm gonna try to walk folks through them on this next slide. And the way that I'm gonna walk through them, I'm not gonna cover all the details in the plots, but what I want folks to pay attention to is at the bottom of the plot, there's gonna be a bunch of different box plots. And on the left, it's gonna represent lower values of that trophic state. So for example, low abundance of oligotrophic lakes. On the right, there's going to be higher values or higher abundance of oligotrophic lakes, for example. The labels are going to represent the variables that are associated with the trophic state classifications. So you can think of this as variables on the right side are more associated with increased oligotrophic lakes. Variables on the left side are more associated with decreased oligotrophic lakes. Okay, once again, don't pay attention to the numbers. There's going to be numbers on there, but I'm just going to walk folks through it slowly. So effectively, we can use all of these data to ask simple questions like, what kind of landscapes seem to promote blue oligotrophic lakes? And what we see is that oligotrophic lakes, by and large, tend to be associated with forest cover, higher forest cover, and also high snow cover. So colder forested environments and the seasonal permanent surface water area. And specifically lower seasonal fluctuation in the lakes. So they tend to be forested and cold and lower seasonal inputs. Whereas places with less oligotrophic lakes tend to have an absence of forest. They also tend to have higher variability, so higher seasonal inputs that might bring in with it nutrients and organic matter, making that lake turn from blue to something else. Okay. Eutrophic lakes. The first variable that pops out is cropland or crops, often associated with elevated nutrients that might go into the lake. But we're also seeing things like wetlands, lower wetland abundance, low snow or this proxy for temperature. Interestingly, we're seeing acid rain or this sort of like this proxy for the processes that cause nutrients to sorb or desorb from sediments as being associated with higher eutrophic lakes. On the flip side, eutrophic lakes tend to be less prevalent, where we have low crops, high snow, so colder environments, and lots of precip, where there might be more flushing throughout the system. But by and large, the main landscapes that promote eutrophic lakes tend to be high nutrients, low retention of nutrients within the surrounding landscape, and warmer temperatures. So I just kind of blew through those, largely because I thought that they were the least interesting of all of them. And these are the groups that, at least at the national scale, aren't necessarily changing as much as this third type of lake type that we're going to talk about, these dystrophic or these browning lakes. And so what kind of landscapes promote dystrophic lakes? Well, they're always in these forested wetland environments. And I'm going to slowly remove the three main mechanisms that seem to be promoting dystrophic lakes. Well, first what I'm going to do, is remove the covering where we don't have high wetlands and we don't have high forests. There's really very little dystrophic lakes altogether. However, a main mechanism that seems to be driving these dystrophic lakes seems to be some interactions between precipitation and residence time, where in particular precipitation regimes that aren't the highest precipitation rates that we see in the US, but are certainly higher, and also residence times that are a little bit lower, less than 1 year, we're seeing elevated dystrophic lakes. So precipitation is one of those main mechanisms driving dystrophic lakes. A second mechanism that the models identified was this interaction of soil organic carbon, and acid rain legacy. as promoting dystrophic lakes. And so what I'm interpreting this as is, it's this acid rain legacy that's ultimately giving rise to elevated browning of lakes. And then the third mechanism that models identified were these lakes with high seasonal inputs, or these high seasonal to perm ratios that we're calling them. And so what I'm interpreting that as a third mechanism that the models identified were lakes with high forests, high wetlands, but also high seasonality, as maybe flushing in organic matter into those systems. Now, we can take these models and ultimately ask questions like, well, where are these lakes within the contiguous US, and where do these mechanisms driving lake browning seem to be concentrated? And so those acid rain, soil organic carbon affected lakes, they seem to be concentrated within the northeastern US and scattered throughout the East Coast, but predominantly in the Northeast, indeed, where we have this history of acid rain. Now, those precipitation-driven brown lakes, they seem to be concentrated in the upper Midwest and to some extent the Southeast. But what this is implying to me, at least, is that lake browning that we're seeing in the upper Midwest is likely driven by changes in precipitation regimes and maybe some interactions with the droughts that have been happening, especially since 2012. Now, those seasonally driven lakes, this was admittedly a bit of a head scratcher for me. Those were concentrated largely in the Northeast, and the Adirondacks really pop out there. And I was a little confused because I was like, this is where the acid rain literature and lake browning literature really has its roots. And then I realized, a gift and a curse of using these machine learning frameworks is that they sometimes show you things that are unexpected. They identify new mechanisms, but we really need that in situ knowledge to really make sense of the models and the scientific results that we're seeing. And so we can use our brains and say acid rain was historically high here. And I think what's happening is that the models are really identifying these lakes as being seasonally driven because they do have high seasonality. But they're also in an area where acid rain was really high. There's lots of soil organic carbon there. And so these lakes, while they're maybe most associated with that seasonality, they're really like those ideal test lakes for lake browning, in that they have the soil chemistry change, they have the seasonality inputs, and they have the organic matter, where they likely give the strongest signal to environmental noise. That's just my take on this. But suffice it to say, they're really just a different form or a clearer signal of that acid rain legacy. So once again, I've shown a bunch of complicated figures at you. And I just want to distill these concepts into a few key bullet points. So what seems to promote different types of lakes at the national scale? Oligotrophic lakes, they seem to be in forested, colder environments, and they tend to have this sort of hydrologic stasis. They're an exercise in moderation. They don't like too much external inputs. Eutrophic lakes, they require sources of nutrients here, like crop cover, but they also need a mechanism to get those nutrients to the lake. So things like less water, less wetland cover, or increased seasonal inputs. And then they also need warmer temperatures. Warmer temperatures allow more algae to grow, and so we tend to see more eutrophic lakes where it's warmer. Dystrophic lakes, they require forests and wetlands, but then there's regional differences. The upper Midwest seems to have interactions between precipitation and, to some extent, residence time. The Northeast seems to have that acid rain legacy, and especially the seasonally driven lakes. Okay, I've just said a lot about what I've done with this data. But as Joe said, there's a lot that other folks might want to do with these data. And so how can you actually access these data and use them for your own purposes? The data are all publicly available with the associated scripts to produce the data on the Environmental Data Initiative. freely available, no paywalls, no registration needed. Yeah, no paywalls, no restrictions. And also our scientific data paper is open access, so you can really get a better sense of how we created the data, and I think maybe more importantly, how we validated the data to know if the data actually meet your specific needs. But just to maybe prime the pump on the types of things that folks maybe focused on an individual water body might want to know, what can you do with those data? So my good friend Tyler King, he's at USGS and based in Boise, Idaho. He's been using these data to show regional managers and individual water body managers about how lakes might change classification when we don't have in situ sampling. So here's the probabilities of being classified in an individual trophic state for Island Park Reservoir in Idaho. And we see that from the 80s to about 2005-ish, the lake was classified as oligotrophic, but it takes a sharp turn after 2000. And we see that eutrophic classification increasing. And effectively, what this can be helpful is that we can understand lake trophic state classifications when we don't have those in situ data. We can also look at landscape scale variation, where especially like in Montana, Idaho, in the northwest, we see lots of eutrophic lakes. And then as soon as we oligotrophic lakes, and as soon as we cross over the mountains into those north temperate plains, we start to see higher eutrophic lakes becoming abundant. So helping understand water quality and ecosystem function at scale. We can also expand these data. One thing that I think is really powerful about this data set is that we've built it off of hydro lakes, that lake shapefile compendium, that has these unique identifiers that allows folks to not only take these data and use them, but also join them with other data based on these hydro lakes shapefile, or these hydro lakes data. And so, for the curious, one thing that I've had the privilege of leading is the creation of the Global Lake Area Climate and Population Dataset, or GLCP, which effectively aggregates a lot of those Landsat-derived characteristics that I was talking about, things like lake area extent, climate reanalysis data, so MERA2, air temperature, precip, long wave, short wave radiation, cloud cover, humidity, as well as snow cover, lake ice cover, and human population within a basin. So you can think about this as just another way where you can not only take your lake and not only identify changes in your lake, but also contextualize those changes with any of these variables that I've got listed up there. Okay, so the last, hopefully, 3 minutes that I want to do is, I've talked a lot here about what you can do with these data and with this science. What I didn't talk about is how do we help people. If you're sitting there and you're saying, I'd really like to get engaged in this science and these data, how do I do that? That's just one thing that I want to offer a potential off-ramp for. There's opportunities like this seminar series or this webinar series that are really excellent for engaging in the aquatic sciences. Another option, another addition actually, is these communities that I've had the privilege of helping lead and co-found, both the Aquatic Ecosystem Modeling Network Junior, or AMON-J, as well as the Community for Data Science and Open Science in the Aquatic Sciences, or DSOS. And what this effectively is, it's a group of about 900 aquatic scientists, many early career folks, who are interested at this intersection of data science, open science, and the aquatic sciences. Real quick, it started all the way back in 2020, where my buddy Jake Zwart and I at the he's at the USGS as well, we created this sort of virtual conference where in the pandemic, and where we started hosting folks that we thought were leaders in the data science, open science, aquatic science spaces. First seminar took off with about 430 registrants. And it's quickly ballooned along with our organizing team over the past 5 years, where we currently have, like I said, about 900 community members in about 70 countries worldwide, particularly the North America and also many African countries. And one thing that I think we've done a pretty good job of is not only showing people what you can do with this science, but also helping create learning opportunities for folks to engage in this science. And so we've hosted several like virtual summits, but also workshop series. And this is, I'm not giving this the credit that it probably deserves. But if you're interested in learning how to work with remote sensing data, with large data sets, with numerical models or data science machine learning models, we have about 75 hours of training material on our OSF repository. And I'm happy to share a link to that in the future. But long story long is if you're interested in not only hearing about this science, but also doing and engaging in this science, there's a wealth of resources there for folks. And so with that, I'm happy to take any questions. Sorry, I kind of rushed it at the end, but I wanted to leave time.
Thank you, Michael. That's that's a lot, and I have to admit I'm still processing. There's so much information that you've shared with us,
Oh,
and it's all really wonderful. You have been a busy guy, so thank you. Um, all right. I do want to get to questions. We've got some questions from folks that are in the audience. So I'll remind folks, if you have questions for Michael, please go ahead and submit them. You can simply hover your mouse over the bottom of your Zoom screen. You should see a Q&A box down there in the middle, and click on that, and you can submit questions. So I've got a couple questions now, and I have a feeling we'll have more coming in as we get started here. So. Um, the first question, I think you've already spoken to it a little bit. I think it was asked prior to you walking through some of the mechanisms, but I'll ask it and maybe you can fill in maybe some gaps in terms of what our mechanistic understanding is. So the question is, how does acidic deposition contribute to lake browning?
gosh. Okay.
So it's
Wow.
not a dissertation level question, but
Yeah, you're
the
making me.
thumbnail.
I am probably not the one qualified to give an explanation. Is Jill Barron, one of the co-authors on this work, definitely is the one to give it. Um, but and please feel free if folks are more knowledgeable than I in the chat. Effectively, what it caused is that it's not necessarily the acid deposition itself; it's the recovery from acid deposition that contributed to lake browning. So basically, you have years of acidic deposition that changed soil chemistry of forests and causing that to acidify. What the acidification ultimately leads is an increasing in the solubility of organic matter. So you have more organic matter that's entering into the sediment. And then what happens is through time, or that can enter water. And then through time, what happens is that organic matter, as it returns back to a basic setting, or a basic environment, it deacidifies. You can have flushing of that organic matter through the system. So just think of it more as like it's really the change in the what's called the acid neutralizing capacity of forest soils that ultimately leads to that lake browning effect. So it's not necessarily the acid rain itself; it's the recovery from the acid rain um, that led to that.
That's a great answer. Thank you.
Well, hopefully I did it justice, but more biogeochemistry folks can give a much more technical explanation than I.
All right. So the next question is, does diffuse solar radiation penetrate the water column better than direct incoming solar radiation, thus increasing photosynthesis and eutrophication?
I honestly don't know. I'm sorry. Um, just I'm sorry. Could you say the question one more time?
Yeah. Does diffuse solar radiation penetrate the water column better than direct incoming solar radiation, thus increasing photosynthesis and eutrophication. So I think the question is,
Yeah. Um, Admittedly, I don't know. Um, and I don't know of any, yeah, I can't think of studies offhand that address that exact question. I do know, I, yeah, I'm not entirely sure, and I don't want to misspeak.
yeah, that's my sense too. That's
Yeah,
a pretty specific and difficult question.
but
So
that sounds like an excellent area for future research.
that's right. Um, all right. So um, let's see. Oh, so we have no questions in the chat now. I do have some questions,
Cool. Yeah,
as
of course.
always. So, but I'll offer a last call to anybody else that's on the webinar. If you have questions for Michael, go ahead and submit them with a question and answer function. So, um, you know, I, here's one before I get going with mine. In the northern latitudes, has there been a significant reduction in ice cover on an annual basis? And if so, how much additional radiation might be expected as a result?
A lot of radiation questions.
Yeah.
Uh, um, so has there been less ice at higher latitudes? Within the contiguous US, yes. Um, like marked decreases. Um, you don't necessarily see the trend at the time scale that we're looking at. So this 1980s to today, you need a much longer time series for that. Um, but at least at the interdecadal or at the close to centennial scale, you do see marked decreases in ice cover. And does that lead to increased radiation to the lakes? Um, yes, sometimes, because because you don't have that ice and that snow on top of ice that might block algal growth. Um, However, there is a wealth of literature starting since 2017 about all the sorts of algae that are occurring underneath ice. Josh Culpepper at York University had a great review paper come out last year about how ice quality, so how cloudy ice is, can influence algal composition under the ice. Um, Shameless plug. I was a co-author on a paper about ecological and societal consequences to lake ice loss. Um, so if you're interested in something in that aspect of lake ice loss, happy to share a link to that.
Thanks. And yeah, we also at the MGLP have had a couple of papers or webinars that we've featured with folks looking into ice loss. So if folks are interested in those, you can also find more in the webinar format on our webinar page. Um, will there be a Hacking Limnology 2025 event? You can run it back?
Oh wow. Yeah, we're. I mean, I hope so. We're currently in the works. Yeah. So one thing I didn't say is that we take great pride in being a community of early career researchers for early career researchers, and many of us at the organizing level are, you know, and entering our golden years of being early career. So we're trying to find the next generation of early career researchers, but we are actively working on having a 2025 Hacking Limno, which would be the fifth one.
Perfect. Thanks.
Mhm.
Um, Question is, can your tracking data access specific lakes?
Yes. Yeah, so it's they are all aggregated at the lake level. For the analyses, we do it at the basin level just because I mean, you're really learning how the landscape is changing rather than why an individual lake is changing. Um, but it is at the individual lake level, but it's only for not only it is, but it's for lakes of at least 10 hectares in surface area. So zero point 0.1 square kilometers, I think. Yeah.
Yep. And we shared the link in the chat for accessing those data, and we'll also share that link through our email with participation or participants as well. So it is 2:58 here in Michigan, 1:58 where many of you are. It's time to wrap up. So I want to just echo the comments that I'm seeing in the Q&A now and saying thank you, Michael, for your presentation. Really a wealth of knowledge, and I think a lot of opportunities for folks to leverage these data in whatever the work is that they do, whether it's research or management, or maybe they're stewards of their own individual lakes. Um, so our next webinar will be next week, same time, March 18th, and the subject is going to be U.S. lakes are monitored disproportionately less in communities of color. So a really important webinar. Jessica Diaz Vasquez is going to be presenting that webinar, so I encourage folks to check that out and sign up for that webinar. Um, and we look forward to seeing you all in a webinar in the future. Thank you again to Michael, and have a good day, everyone.